Hello, welcome. I'm Huang, a staff scientist from Computational Biology Branch, National Library of Medicine, National Institute of Health. Today, I'd like to share with you the result of my recent study about the role of super silencers in the carcinogenesis and human development, with particular interest in the B cells.
So, can you either speak louder or closer to the microphone? I think the room is big enough that the people in the back are. If you can move it any closer to you.
But is this which one is working? I don't know.
This one.
Okay. The silencers are DNA sequence, which recruit the repressor and loop to the target promoter, subsequently downregulated gene expression. They are categorized by the repression histone mark, such as S3K27ME3. Silencer and enhancer work together for the process control of gene expressions, turning the gene off or on at right time and place. However, as you can appreciate from this plot, the number of the publication over last 2 decades, silencer overshadowed by enhancer. Partially due to the experimental difficulty in silencer characterization. In past several years, we developed the machine learning, especially deep learning model to predict the silencer with high accuracy. Across this,
We can see the laser pointer. Is that what you want?
Yeah. Okay. Then okay. I do it over here. How I control this laser pointer?
You are controlling it, right?
With this one?
Yeah, we can see it on the screen.
Oh, okay. Across the several cancer line, we observe the tendency of the silencer clustering into lung formation, reminiscent of the enhancer forming super enhancer. This clustering phenomenon is evidence in the distribution of H327ME3 in intensity, which is similar to the intensity of the H3K27AC. Both have the long. Let's be confused. This I don't know how to control. I mean, yeah, both have the long right tail, indicating the existence of the large clustering.
There seems to be two things happening at once.
Yeah, I feel like. I know it's. I don't know which one is my control.
Excuse me, I cannot hear it like this. Just speak louder.
Yeah, we can do that. We can bring the. So there are two things that are happening on the screen. One is this laser pointer that someone else seems to be controlling on.
I don't know. This one doesn't have record. Yeah.
Here. Laser pointer.
No. But where that's the other one from? Yeah, where that from?
No idea, really.
It might be a recording.
It might be something, but yeah.
I think I deleted all the recording.
No, it is saved on the slides. It looks like.
Oh,
Yeah.
okay.
You could go back to this.
But there's nothing.
Go back here. No.
Yeah.
No, definitely recording.
Yeah.
I will do this.
That'll work. Yeah.
All right, and then we can go back. Ideally and hopefully now you should be able to.
Yeah.
Let me see if I turn laser pointer. Okay, you can move it around on this screen, and it will show on there wherever you're showing.
Okay.
And then you please be closer.
Okay. Yeah, I will. I will closer to this microphone. Yeah, to the speaker. And so all this observation purpose us to use the super enhancer detection framework to the silencer data, leading to the identification of the super silencers in human genome. Approximately 900 super silencers were detected in the B cell line 12878 with a comparable number of the super silencers predicted in other cell types. To evaluate the activity, of super silencers, we first examine the expression of their associated gene, and those genes are downregulated, and also their downregulation level more pronounced than that of the gene associated with the typical silencer. In this presentation, I will use the abbreviations SS and SE represent the super silencers and the super enhancers. S and E represent the typical silencers and enhancers. In our next validation step, we use the ATAC star-6 scores, which measure the strength and also directionality of a regulatory impact by genome region. So those scores are forming the robust silencing effect of the predicted super silencer, surpassing the upregulation level induced by super enhancers. We also accessed the super silencer activity using the lymphoma-based silencer assays conducted by our collaborator, Dr. Laura Elniski from NHGRI. Laura's lab demonstrates the 4 components of BARC2 super silencer in B cell downregulate the significant downregulate reporter gene in B cell, but not in K562 cell, indicating the super silencer activity and also tissue specificity of the predicted super silencers. After validation, after these validations, we want to explore the connection of the super silencer with the carcinogenesis. While examining the distribution of B cell cancer variants, we noticed the hypermutation chain within super silencers. For example, as depicted in this plot, the density of GWAS super B cell cancer variants rise sharply in within the super enhancer and the super silencer region, indicates the pivotal role of those elements in carcinogenesis. Also, the super silencer variants are more likely associated with multiple B cell cancer types. The recurrent nature further supported by the patient data. This table lists the genes proximal to the top hypermutated super silencer in B cell cancers. This list is notably enriched by the master regulator of B cell development and differentiation. In addition to hypermutation in cancers, regulatory landscape of the super silencer are frankly disrupted by the cancer translocations. It's about 4 times as often as expected in lymphoma. Notably, 26% of super silencer translocation merge those elements with super enhancer domain, far surpassing expected 10%. The illustrative example of super silencer translocation is BCL6 translocation. This translocation is a hallmark of diffuse large B cell lymphoma, DLBCL. 20 of 23 BCL translocation breakpoint, 20 involved the BCL6 super silencer, and 17 those translocation merge the BCL super silencer with a super enhancer domain, either in ROHA or IgM. Both of the genes are highly expressed in mature B cell. The BCL6 it's a gene with the developmental significance, highly expressed in development stage. We demonstrate that the super silencer downregulating the expression of BCL6 in mature B cell, and this state reversed in the B cell cancers. In order to explore if this kind of the downregulation and the reversal is the common function for the super silencers, we examine gene expression in 4 cell types. There are H1 embryonic stem cell, mesoderm cells, hematoprogenitor cell, and mature B cells, representing the key development stage of B cell. As you can see, the gene proximal to the B cell super silencers, so lowest expression level in mature B cell and highest expression level in stem cells, as depicted by this blue line. This chain suggests the large transformation of developmental super enhancer into the mature B cell super silencer, accompanied by the opposite chain, which represents developmental super silencer morphing into mature B cell super enhancer, as illustrated by this red line. The function of the B cell super silencer downregulating developmental important gene is further supported by the gene etiology analysis based on the grid, which highlight the several embryonic development stage associated with B cell super silencer. The functional switching between the super silencer and the super enhancer is not surprising given our previous publication on the large-scale functional conversion in the human genome involving the silencer-enhancer transition during the development and between the cell lines. In this study, we observed that the 30% of B cell super silencer are converted into super enhancer in DLBCL, that's the lymphoma, B cell lymphoma. And also the 22% of this conversion reoccur in more than half of the profile, the DLBCL cases. About the 2 times as often as expected, suggesting the super silencer to super enhancer play the crucial role in carcinogenesis. In order to further understanding the impact of super silencer to super enhancer conversion in carcinogenesis, we examined the BRT4 concentration in DLBCL. We found out the BRT4 saw the highest concentration level in the super silencer, which converted from the B cell super enhancer, which converted from B cell super silencer, suggesting that the super silencer to super enhancer conversion reinstalled the most potent developmental super enhancers. We also examined the BRT4 concentration after GQ1 treatment. GQ1 is the strong inhibitor of the BRT4 and effectively wash out the BRT4 concentration along all the super enhancer region, leading to the downregulation of their target gene. The gene associated with the conversion, super silencer to super enhancer conversion, so the most significant downregulation level. This level is about twice as pronounced as the gene associated with all super enhancers, and about 4 times as pronounced as the one is expected by chance. The BCL6 is an example of the gene associated with the super silencer to super enhancer conversion. So our study demonstrated that the B-cell super silencers inhibit activity of the developmental super enhancer and effectively shutting down the gene, their target the developmental gene. And in the B-cell cancer cases, this regulatory effect is reversed, likely by the sequence somatic mutation or translocations, and which leading to the reactivation of the developmental gene in B-cell cancers. In summary, our study saw the unique function of the super silencers in B cell. They are contribute for the established and maintain the B cell identity. And also these elements are hypermutated and frequently translocated in B cell cancers. Finally, we report the super silencer to super enhancer conversions as one of the mechanisms underlying the carcinogenesis in B cell. So finally, I want to acknowledge all my collaborators and also my colleagues. And I want to now mention that our funding from the intramural research of funding in National Institute of Health. Thank you. Thank you for your attention.
Just accommodate one quick question. If anyone has it, you don't even need to come here. You can ask, and then speaker can repeat.
I have a question. Is when you mentioned about the super silencer being enhancer, so in one cell stage, the silencer always being silencer to every target gene, or they will kind of enhance some gene, but a lot of them.
In one cell type, in one, or I shouldn't say in more precisely, in one biological context, this only work for the super silencer. But when the cell type context change, then it may transform into super enhancer.
So the enhancer or silencer in a cell is always work as a silencer, no any effects.
I shouldn't say. He said there's the one genomic region in one condition could work, have the bifunction, you mean, could work in the either way, in super as a super silencer and also as super enhancer. I shouldn't say it's zero case for that but we didn't find too many. Yeah, I shouldn't say definitely no. I cannot say that. Yeah, but that's definitely not general cases. Yeah.
Let's thank the speaker.